So I'm Daisuke Kihara. I talk about how to now evaluate the protein structure models. But here I want to consider that proteins are not rigid but flexible. So, there are lots of occasions that you compare protein structures. And in many cases, you superimpose two protein structures, considering that two protein structures are rigid. Typically, if the chain, length of the chain is identical, you compute RMSD value. Basically, RMSD value is computing after superimposition distance between corresponding points, maybe the C alpha positions, and then kind of average it. Also, if two protein structures are not does not have same sequence lengths, then you need to use different methods. For example, CE, DALI, SSAP. Well, if you're interested, we also have a very fast and interesting methods to search, compare protein surface of proteins. But all those methods are dealing with the rigid structure of proteins. Here. And also protein structure comparison is very important for evaluating which structure computation structure model is more closely, correct to the ground truth of the model. And for that, there are methods, scoring function called TM score, which Professor Shingao has introduced in the previous talk. And then also there is a scoring function called GDTTS. Well, what GDTTS computes is after superimposition, it basically counts number of amino acids within 1 angstrom, 2 angstrom, 4 angstrom, 8 angstrom, and then compute a fraction of it, and then basically average it. But all these methods are using the rigid protein structures as a reference. Because PDB provides the rigid structure and it's easy to use. And we are very used to look at the very static picture of protein structures. However, as you know, proteins are flexible. Here in this picture, I'm showing how the NMR determined structure can vary. And also here, if you run 10 nanosecond molecular dynamics simulation, sometimes the tail region can move that much. So, and the flexibility can be obtained by NMR or molecular dynamics simulation by computational methods, or more coarse-grained molecular dynamics simulations using, for example, Gaussian network model or normal mode analysis and so on. And actually, there are also very interesting discussion that even the X-ray crystal data has a very flexible information, which is not just limited to the B factor you can see at the rightmost column in the PDB file. And well, proteins, well, there was also lots of very interesting extreme cases that proteins even do not have a rigid structure at all, which I cannot deal with it. This is because too flexible to me to compare protein structures, or maybe not meaningful even to compare protein structures. But, well, the message here is protein is flexible. Actually, people notice it, of course. And then there are several methods to compare protein structures using flexibility. Obviously, when you compute RMSD value, you can give a weight to individual residues by considering, for example, B factor, or you can run RMSD computation iteratively and then use the distance taken from the previous iteration as a weight. Well, this is FlexE methods by Kendall's group. Used a Gaussian network model in evaluating the structure in terms of energy, but not the structure, which is an interesting idea. And there are also methods which compares two ensemble of structures taken from NMR or MD simulations, and then compute, for example, KL divergence, Kullbach-Leibler divergence. Or there is also methods to use some maximum likelihood methods to compare a structure into ensemble structures. Now here, we developed a scoring function to compare computational structure model to the crystal structure, but considering the flexibility. So we make the ensemble of structures from MD simulation and consider that ensemble as a ground truth of the computational model. Ensemble can come from NMR or some other methods, but here I used the 10 nanosecond MD simulations with explicit water. Okay, so here, since I have the ensemble of structures, X, which contains many structures, as one of the structure X of I can be represented in this way. So X of I is deviating with E, the vector from the mean structure, and it should be rotated for the superimposition, and it should be shifted. So first I need to superimpose an ensemble of structures all together. And this is a bit complicated, but I'll tell you the story how it is working. So now here, okay, for each of the structures, I need to determine the mean structure, average structure, and then rotation, and then translation. So I need to maximize, I need to optimize these three values. To do that, first I assume that this is sigma is a covariance matrix which describes how the ensemble structures in ensembles are deviating from each other. But first I assume that the covariance is 0, which are identity matrix, which means that all the structures are identical, there's no variation at all. And then also assume that one of the structure is equal to the mean structure, just as a starting structure. And then this alpha value, this is a parameter I explain later. Now, once I assume the mean and the covariance is 0, then I can compute the translation vector to superimpose. And then once T is determined, then I can compute the rotation of individual structure and then estimate the mean structure. Once I get mean structure, then I can recompute, optimize the rotation and mean and iteratively. Now, assuming that all structures are identical, I have the translation, rotation, and mean structure. But now, on the other side here, what I'm doing is to actually compute the covariance matrix, now to consider the structures has some variations, ensemble has some variation, the individual residues are moving in the different ways, different degree. To do that, okay, first I can compute covariance matrix because it's already superimposed. And then, well, of course, I can compute eigenvector. But here, so from this iteration is to optimize this covariance matrix, assuming that the eigenvector of the covariance matrix is just one assumption that the eigenvector is following inverse gamma distribution, which basically what I'm trying to do is that residues which does not deviate much have a very small probability, and also the residue which deviate a lot also have a small probability, and they have some distribution. And then after I compute the covariance matrix, then I come back to this estimation of M, R, and T, and so on. Now, okay, once I'm prepared now I have superimposed all the structures in ensemble all together. Now, okay, that's a grand truth of the computational structure model. Now I have suppose I have a computational model from somewhere. Then I want Y. Now I want to evaluate how good this model is relative to the ensemble of the structures. To do that, I rotate the model based on the covariance matrix I computed before, and then very simply, flexibility score of model Y is basically the difference between the superimposed Y to the mean of the ensemble structures, and it is weighted by the eigenvalue of individual residues. If the deviation is very large, eigenvalue is large. So if the deviation is very large, if it is a tail region which is very flexible, even if the residues are in the model is deviating a lot, I don't care because it's already flexible. But if the residues are very solid, it doesn't move much, then small deviation will cost some, is penalized. And it's averaged. I can also compute the flex score in the GDT-TS-like score. Here, I just, after superimposition, I count the number of residues at flexible score of 1, 2, 4, 8, and then compute the fraction, average fraction. Flex score is, best flex score is 0, because that means that that model does not differ from the mean, and then it will become, it's basically telling you how different model is from the mean of the ensemble. Okay, let's see how it works in the toy model. So these gray chains are taken from NMR structures models from PDB. As you see that there was a large deviation in the flexible tail region. And actually manually, I constructed two models, blue and green. And actually, this green model and blue model have identical RMSD value, GDTTS and TM score. I made it in that way, actually. So in terms of GDTTS, for example, these two models are identically correct, or identical, have identical quality. But I would argue that, and hopefully you agree, that blue model is better because the deviation of the blue model is at the tail region, but in contrast, green model have a large deviation from the alpha helical regions. So this is represented by the flex score. The first panel is showing how much each amino acids are deviating within that NMR models. So there was a large deviation in the tail. Now the second panel is showing the distance of each model. Green one has deviating in the middle of the model here. And then both model, green and blue, is deviating from the mean of these NMR structures, which is shown here. But if I compute the flex score of individual residues of blue and green model is penalized in this region with a large, bad flex score. But the tail are not penalized because it's already within the range of flexibility, and I don't care because it's flexible and it's in the range of the natural flexibility. That's a concept, and basically that's a whole message here. But I can now see how it works, how it look like if I apply it to the CASP protein structure models. So here we took several targets in CASP, and then for each of them I downloaded more than 200 prediction models from CASP website. Then I'm just computing correlation of those models between the flex score we developed, and then, for example, RMSD value, TM score, and GDTTS of the 200 models. First, RMSD have very good correlation overall. TM score and GDTTS have overall good correlation in many cases, but you can see that sometimes it has very low correlation. This happens typically when these three are the average value, average score of the 200 models. This very weak correlation or no correlation occurs when the model are very bad. If the model is very bad, well, it has a very large discrepancy. Now, okay, because RMSD value and the flex score has a high correlation, but actually to show that actually they are not identical, I'm showing several examples here. So y-axis is showing the correlation coefficient value between flex score and then RMSD value. X-axis is showing how much each residues are deviating in the MD simulations. So here, these are the several targets, each of which have 200 models, where the RMSD and flex score have very high correlation, close to 1.0. This happens when proteins are not flexible. It's very natural here. For example, this case, 714 here, like here. This is the residue position of protein target 714. Y-axis is showing the standard deviation in the molecular dynamics simulation. They are fairly stable. Well, even if I use flex score, if protein is flexible, protein doesn't move much. So naturally, it has a very close correlation, high correlation with RMSD. That's what's shown here. But if proteins have large flexible region like tail regions or in the middle like this, then flex score and RMSD start to deviate in some degree, because even the model which is not close to the crystal structure may not be penalized as much by FlexScore if the flexibility is coming from flexible regions. Now I show you some little bit more example. So this is another target, and then I computed the FlexScore, GDTS, TM score, RMSD of those about 200 models. Well, you see that overall it has some reasonable correlation, but you see some differences here. For example, this region. These are the models which have variety of RMSD value, but the FlexScore is kind of stable. And these are example of two models like this. So this, this is orange, but the red and green models have some difference in the tail region and this region. FlexScore says these two models are similar quality, 2.75, 2.71, because deviation is at the tail region. I don't care. It's in the range of the physical fluctuation. But the RMSD value, obviously, penalize, consider this because this is deviating from the mean of from the crystal structure. Another example here, these are the example when the TM score, or let's say GGTS score, have very different score. Same score, but the flex score have several very different values. So this case, here the green one and orange structures have a similar core structures, well predicted core regions, and that's what GDTS score is picking. So that's why GDTS score is similar, 0.52, 0.51. But the flex score does not like this long tail, strange tail or loop regions, which is far away from the ensemble. That's why flex score prefers green one over orange one. A similar story here. Another target. You see some deviation. GDTS score is similar, but flex score does not like one from others. This is an example. Orange and green has similar GDTS because the core regions are modeled well, but the flex score does not like this green tail because it's too far away from the ensemble. This is more subtle example here. This is a beta sandwich structure where you see the two models are capturing the topology very well, but you see that green model is deviating a little bit here, these regions, from the ensemble. There was some deviation in the orange model here, but overall, green model have more deviation from the ensemble structures. So although GDTS have a similar score, flex score prefers orange one because orange one does not deviate that much as the green one. So this is another discrepancy between flex score and GDTS. Now, okay, you might wonder what will happen, because this is MD based. Well, if I run MD, use different MD trajectories, obviously the score changes a little bit. So here, in these two examples, I ran MD simulations from the crystal structure 3 times and see how the MD simulation differs. There were some differences. It's true that flex score will be affected by those small changes, but it's not too much. Same message here. There is some deviation from the 3 trajectories or from MD, but in this case, one of the model have identical flex score. This is showing how the flex score depends on the length of the MD. Correlation does not change that much from the flex score and MD. This is showing robustness between this MD-based ensemble from NMR ensemble. They correlate very well. Now, okay, now if I apply this scoring function to the CASP groups, what will happen? So this is a ranking by flex score. This is GDTS and TM align and so on. You see that actually top groups, A, B, C, are fairly stable. There are some variations at the bottom of the ranking. If we want, we can also predict the flexibility. We have a server or FlexSpread which compute the deviation of of the fluctuation of individual residues using the support vector regression rather than using the MD simulation. Okay, so summary is that we developed FlexScore, which considers flexibility taken from MD or NMR. It distinguishes discrepancy at the core region and then tail regions. Overall, it correlates very well, but there was some discrepancy from between FlexScore and conventional score, but I believe FlexScore is reasonable. And thank you very much. This is the Michał Jamros, who did the work when he visiting us from his university. Thank you very much.